Now we're going to continue on with section 1.3 today and wrap up chapter 1. Now 1.3 is going to focus on how do you ultimately go through the process that we would consider experimental design. So whenever you're trying to test something, you need an experiment to test it. So we're going to focus on what are the parts of a good experiment. We're also going to talk about how you represent and analyze the data that you get from an experiment. So starting off, let's go over some terms to make sure you're comfortable with them. So when you're doing an experiment, one of the first things that we typically want, this is what is present in most good quality experiments, is a control group. Now the whole purpose for a control group is to have a group of individuals that we are going to not test. We're going to kind of leave them normal. We're not going to manipulate them. And we're going to use them for comparison. So for instance, if I think coffee stunts your growth, makes you shorter, I need to know how tall people are on average that don't drink coffee. So that way I can have an experimental group, that's going to be these guys, the ones that you manipulate, where I might say, all right, I'm going to have a group of these guys that drinks 1 cup of coffee a day, maybe another group that drinks 2 cups of coffee a day, and I'm going to analyze their average height. And then I can compare their average height when they drink coffee, depending on how much, with what it would have been most likely if they didn't. So now I can get a sense of, are they actually shorter than they would have been, or are they taller, or are they about the same? Now, some labs will not lend themselves to having a control group. Sometimes basically everybody's being experimented on. But if possible, if you're able to have a control group, you should always try to have one. That way you can set a baseline so we can figure out exactly how much something changed. Now next we've got a series of variables. So the independent variable is what you are going to change. So this is sometimes referred to as the cause. This is ultimately what the experimenter is going to be changing. So in our experiment from up here, that would be like the amount of coffee you drink. So this is independent of everything else. This is just up to me as the person designing the experiment. I'm going to say, all right, you drink no coffee, you drink 1 cup of coffee, you drink 2 cups. It's under my control. Now, the fact that people are drinking different amounts of coffee should cause some effect. And that effect is going to be our dependent variable. So this is what I'm going to measure, and that's what I'm going to put down here. This is essentially what's being measured to try and see what my change, the independent variable being changed, what effect that had, what it caused. And so in this scenario, the one that we did with coffee, so this would be like coffee, okay, coffee drank, and then below it, our measurement was going to be height. And because you do this with a lot of people, you'd normally take and average that. Because we can't just look at one person, we need a group. So that's going to be the basic ways that variables work, at least for our 2 main ones. Independent variable, what are you going to change? Dependent variable, how are you measuring the effect that that change had? Now, constant variables are typically every other variable in a lab. And that constant variables are just ones that are unchanged. So if I'm trying to do the coffee experiment, I would want to make sure that there's not other things going on that affect their ability to get taller. So I'm trying to make sure that they don't get injured. I'd ideally like them to drink the same amount of water, to drink the same or eat the same types of foods in the same amounts. So ideally the rest of the variables that we're not experimenting with, we try to keep them the same, constant, unchanged. So those are oftentimes referred to as constants or constant variables. Now, confounding variables are ones that we know can mess things up. So these are things that could occur, like we talked about, where maybe some of them are eating some other food that's known to cause stunted growth. Maybe some of them aren't eating enough food, which could stunt your growth. Maybe some of them had injuries. So these are things that ultimately can, and not being as elegant as perhaps I could be, I'll just say it could mess up our data. So these are variables that could affect our dependent variable. They could affect our measured results, but they're not what we were studying. They're not the independent variable. They're different. So mess up our data is ultimately what these guys can do. And so a lot of times you'll see someone say that in their experiment that they adjusted it to remove confounding variables. So if they knew some guy ate not enough, they might take and either remove that person from the study or they might adjust his height to be higher, so to speak, to adjust for the fact that some of that stunted growth was because he didn't eat enough. So examples, I just want to make sure you guys can identify if I give you a hypothesis or a setup, I want to make sure you can pull out what is the independent variable, what are we changing ourselves, and what's the dependent variable, what are we measuring. Now, A lot of times when we do a lab setup, you'll see this if-then statement. That's what this first one is. So if you spend more time studying, then your grade will improve. So what are we first changing? And that's the if part. So the first thing we're doing is spend more time studying. So the if you spend more time studying, that's our independent variable. Now the dependent variable is what's changing as a result. The idea here is we're trying to figure out that the grade will improve. That's what we're essentially measuring, is the grade. Now for the second one, we have higher temperatures cause bacteria to grow faster. So what's coming first, the higher temperatures or bacterial growth? And in this case, hopefully you realize it's the higher temperatures that are causing this. So the higher temperatures are what's coming first. That's what we would have done. We cranked up the heat on those bacteria. So the independent variable then is the high temperatures. Wow, that was drawn terribly. And then the bacterial growth is ultimately what's going to be the dependent variable. That's what we're trying to measure. Now, a hypothesis is going to be this prediction, this statement of what we think is going to happen based upon what we know. And you don't have to write a hypothesis as an if-then statement, but especially starting out, to make sure you don't screw it up, because sometimes you try to throw too much into a hypothesis and you can make it where it's not valid. It's very easy just to use this if-then statement idea. So the if part will be our independent variable, as you saw from before, and our then part is our dependent. So if more fertilizer is used, so we're changing, we're controlling in the lab how much fertilizer is applied, then the plant height will increase. So we're trying to find out, we're trying to measure plant height to see how it's affected. Now, the other key part about a hypothesis is it has to be testable. You can't just say, I think that ultimately if I think really hard, a planet that we can't possibly detect will blow up. If I can never check to see if that's accurate, if I can never test what you're telling me, it's not a hypothesis. I mean, it ultimately is going to lie outside the realm of science, at least until we're able to test it. So make sure your hypothesis is something that we can actually test.